Hi, and welcome to our first recorded lecture. So in this lecture, this will be kind of just the very intro material that you need to know really to help you throughout the rest of the semester. To get us started, there are some things that I'd like you to know in general just about all of your recorded lectures. So because this is an asynchronous class, your lectures will always be recorded, which means that you can watch them on your own time. Another great thing about this is that you can always pause if you run out of time or need to go do something. You can rewind if something didn't make sense and you want to spend more time with it. So really use those recordings to your advantage. I will also always provide you a student version of the PowerPoint that I'm presenting off of, or if I've asked you to watch a YouTube video, I'll create a note catcher, like Word document that you can also use to take notes. You are totally welcome though to use a notebook and take your notes completely from scratch, whichever you prefer. In terms of just kind of the general class, my goal is for you to have about 90 minutes of like quote unquote lecture each week. I will break that 90 minutes into smaller videos just so that you don't feel like you have to watch a 90-minute video from start to finish. So you'll see that I give you multiple lectures to watch each week, but my goal is that they never total more than 90 minutes. If they do, I'll always kind of give you a notification about that. And then that way, the rest of the week is just for you to work on your assignments that you need to complete, be that a discussion, your lecture, or any other lecture assignment, or your lab assignments. You'll also see that every week I will give you guys practice assignments that help you review the material that is covered in all of the recorded lectures that you're provided. It'll also help you study for those quizzes. Basically, we have quizzes every 2 weeks, so roughly every 2 or 3 chapters you'll be taking a quiz. So those practice assignments can really help you to review that material and kind of stay on top of things. So that's really the general kind of idea for all of the lectures throughout the semester. This video specifically covers some of the more introductory information that you need to basically understand the rest of the semester. So in this video, we're going to be covering the basics of all living things. We'll see that all life has some defining characteristics that they share. All organisms share these things. And we'll also be talking in detail about the scientific method. Next week, week 2, you will have a lab that's designed all around the scientific method. And so I wanted to introduce this material to you this week so that you can kind of start preparing and thinking about how will you use the scientific method in your lab next week. But let's go ahead and start with the basics of all living things. So the very first section, section 1 of chapter 1, really covers the fact that all life has 7 common characteristics that really define all living things. And so we're just going to go through those 7 characteristics kind of quickly. So first off, all living organisms contain critical macromolecules, which we'll be talking about this week as well. There are 4 macro or large molecules that are necessary for life, and those are the nucleic acids, that makes up our DNA and RNA. There are the lipids, or what we more commonly refer to as fats, proteins, and carbohydrates. All living organisms need all 4 of these molecules in order to create cells, in order to perform chemical reactions, in order to control kind of their day-to-day functioning. So these 4 macromolecules are very, very important for life. And when we get into our week 2 material, we will see that these molecules are largely important for creating cells. And that is another common thing that all life shares. Every living thing on this planet has at least one cell. So living things have to be made of cells, or at least one cell to be considered alive. And so we can really think in biology as the cell being the basic unit of life. This figure here on the left shows different types of cells that we have in our body that we'll talk about in detail. Red blood cells in red here, and white blood cells that are white or kind of bluish colored here. So there are lots of different types of cells. Specifically in our body, we'll see that cells are very specialized to do very distinct jobs. However, all living things are made of cells. There are some organisms that are unicellular, meaning their entire kind of makeup is just one cell. Or we can be multicellular, like us. We are made from hundreds of thousands, if not millions of cells that function together. So all cells have these all living organisms have these critical macromolecules. All living things contain cells. In addition to that, all living things are able to grow and reproduce. So you've experienced this in your own life. You have started out as a tiny little child, and then you've grown into the adult kind of body plan that you currently have. And all organisms can reproduce to create new life. Some organisms do this by a process called cloning, meaning that they literally make an identical copy of themselves, and that becomes their offspring. But other organisms do so by sexual reproduction, which means that in this type of reproduction, two organisms need to mate, and they will share their genetic information to create offspring that are not identical to either parent. But in either type, cloning or sexual reproduction, the whole goal is that organisms are reproducing new life. They're creating a new generation that can go on to create a further generation, and so on and so forth. So being able to grow and reproduce is a very important characteristic that all living things need to have. All living things also have very specific energy requirements. We have to eat every day. We need water every day. These materials that we eat allow us to have energy to perform metabolic reactions in our cells, but it also allows for us to grow new cells, to heal damaged tissues. Really anything you can think of that needs to happen in our body requires energy. So we do need raw materials in which to perform those chemical reactions. So we have these macromolecules, we have cells. All living things are capable of growing and reproducing. All living things have very specific energy requirements. Living things are also able to respond to their environment. That can be, organisms can respond to each other. So an organism can respond to another organism, or living things can also respond to changes in their environment. When we're all kind of stuck at home, working from home right now, right? And so you're probably interacting with other members of your family, unless you live alone. So that would be an organism to organism interaction. Maybe throughout the day, the temperature in your house changes and you have to put on a sweatshirt or maybe take that sweatshirt off. In both cases, you're responding to changes in your environment. You're responding to a person appearing in your environment or factors of your environment changing. We will talk about this key term a lot as we go through really all of the organ systems in the body. But one of the key things that all living things must do is maintain homeostasis. Homeostasis, so stasis is kind of a Greek term for state, and homeo means same. So all organisms have kind of this state or kind of way of existing that they need to maintain. For us, for humans, we can think of homeostasis specifically with our body temperature. We need to keep our body temperature at about 37 degrees Celsius, which is 98.6 degrees Fahrenheit. If our body temperature drops or rises above that, we have specific reactions that happen in our body that allow us to get back to that temperature. We'll talk a lot about the fact that homeostasis is actually maintained through a series of chemical reactions. And these reactions can be used to maintain our body temperature. If we get too hot, for example, we sweat. If we get too cold, we shiver, but either way, we're trying to regulate and bring our body back to that 37 degrees. We also have chemical reactions that help regulate our fluid balance. So this is where, when we talk about the kidneys and the kind of waste management system of our body, we'll see that our body is doing a lot to maintain fluid balance. We also want to maintain healthy internal environments. We want to make sure that we have a right enough amount of water or salt balance in our cells and things like that. So really, our body's working pretty constantly using the energy that we obtain through the food that we eat to maintain kind of this healthy level of homeostasis to keep us alive. And then finally, the last big characteristic that we'll talk about that all organisms share is this idea that all organisms are capable of a process called evolution. What that means is that all organisms can adapt to their environments and they can evolve. This is a very slow, very, very slow process, and usually happens from one generation to the next. But in general, what evolution is that all organisms living in their current environment have a favorable trait. For example, maybe there are some people currently who have a natural immunity to COVID-19. I don't know if this is true, but let's just say that there are some people who they cannot get sick. So here's our person. They cannot get sick from COVID. That means that they have a natural immunity. Well, right now, that is considered a very favorable trait. That's a good thing. And so if this person was to meet up with another person and reproduce, it could potentially pass that trait on to their offspring. So we'll do a little curly cue of hair on the top to show that this is a baby stick figure instead of an adult. So how evolution works is that certain organisms have traits that are favorable. And if those traits exist in the DNA of a person, they can pass that trait on to the next generation. And so now that person also, that baby also now has, let's say, COVID-19 immunity. And then eventually when that baby grows up and reproduces for itself, it could also pass that immunity down to the next generation. So what happens is from one generation to the next, we're causing a very gradual change in organisms over a very long period of time. Think about it, a generation of humans can be anywhere from like 80 to 100 years old, right? We live that long. So it's a very slow process, but over time we can change the way that our traits are, or what types of traits exist in a population. For example, in this picture here, we have orchids that have evolved to live in like the branches of trees. There was probably an orchid that before it lived in the trees lived on the ground, but it realized that, oh, if I find a way to kind of climb and grow up this tree, I can get closer to the sunlight and I can perform more photosynthesis and I can survive better. And so from one generation to the next, maybe these orchids got better and better at climbing these trees until finally they just grow on the trees and don't have to start at the ground. So evolution is a very gradual change that takes many generations to really see a big shift in how populations of organisms function. However, all organisms are capable of evolution. That's a big thing to kind of keep in mind as we go through our class together. So all life has these 7 characteristics. Additionally, biology as kind of a topic is a very large topic. And so in order to kind of better understand the different types of science that can be studied in biology, biology is often organized into different levels. So this kind of information is covered in chapter 1.3 of your textbook. I do want to highlight that this figure that I'm showing you here is different from the figure that is shown in your textbook. So overall, what this figure is showing is that life can be organized from the simplest to the most complex. How your book does it is it calls molecules level 1. And it says we have molecules, which we're going to talk about this week as well, that come together to create structures called organelles, which would be level 2. Organelles work together to create the next level, level 3, which is cells. And then we have cells working together to create tissues, which is level 4, and then organs, which is level 5. This is really where we are going to focus on these first 5 levels. And by studying these 5 levels in Bio 104, this helps us understand the 6th level, the organismal level. And then in other classes where you talk about like groups of organisms and then different groups working together, that's when you talk about levels 7, 8, 9, and 10 as they are arranged in your textbook, where we talk about how groups of organisms can then interact with other groups, which then interact with kind of the plants and the other kind of aspects of their environment, and then we can actually describe the entire biosphere kind of on a planet-wide level. But notice that biology is organized into groups where we are starting at the simplest, so our molecule level, this is really the simplest level that we can talk about with biology. It's where we're going to start with our lectures this week. We can't really understand how any of these higher levels work unless we understand some basic chemistry of molecules. And then as we go up in these levels, we work our way to the top level, which is the most complex level, the biosphere. And this is really talking about kind of how organisms really interact with their planet. So we're going to really, again, focus on levels kind of 1 through 6 here. Everything from molecules to the organism, and specifically our organism are humans. But if you take other biology classes, you might get into those higher levels of organization. But really, something we're going to see as we move throughout the semester is that biology really builds on itself. And so in the next few lectures that you'll be listening to or watching this week, we're really going to start at that simplest level. We're going to understand what are the building blocks that are required for life, so that we can better understand how all of the higher levels of organization work. But with that, where I'd like to move with this lecture is I'd now like to discuss the scientific method. So we're going to move into kind of the second part of our video. This would be a great time to pause if you need a bit of a break, and you can pick up at a later time for the second half of this video. But let's go ahead and talk about the scientific method, because really for the rest of the semester, a big key thing that I hope you take away from this class is how to think critically and how to really approach the material that you're presented with a scientific mind. And really, that all starts with asking questions. So we're going to talk about this entire process where we start with an observation and we really work our way through to working through an experiment, coming up with a conclusion that allows us to basically answer a question. But really, you truly can perform the scientific method every day. As you walk through and move through your environments every day, you're probably making observations. Those observations can then allow you to form a question. And what we're going to really talk about it is how can we, once we have a question, create a prediction that allows us to perform an experiment to reach a conclusion so that we can actually answer that question that we came up with. But a really key thing to keep in mind when we talk about the scientific method is that there's actually 2 types of reasoning that we can use in order to make the predictions that we need in order to perform an experiment. So let's talk about that. Let's talk about reasoning. So there are 2 types of reasoning that can be used in science, and these are called deductive reasoning and inductive reasoning. Basically, what these 2 do is they start from different ends of the spectrum. So deductive reasoning for general principle. So let's start with this general idea. All men are mortal, meaning we have a kind of certain set of days attached to our lifespan. So that's our general case here. All men are mortal. We can then make a observation. Jason, who was a Greek, kind of from Greek mythology, we know that Jason is a man. So in this case, we have a general principle, all men are mortal, and then we have a specific case. If we know that Jason is a man and that all men are mortal, we can then use deductive reasoning and apply this general statement to the specific case, which means we can come to the conclusion that Jason must be mortal. Another example of this is, let's take the general case that we know that apples are fruit. Okay, so I know that apples fall into the fruit category. That's that's supposed to be a G. Sorry, let me erase that really quick. General statement, right? That's my general statement. There we go. That G looks a little bit store. And I have a green apple. So I just bought a bunch of Granny Smith apples from the store. Okay, there's my apple. So I know that a Granny Smith apple is an apple. That's my specific case. Into that, here's my specific case. I have a green apple. If this green apple is an apple, I can make the assumption that this green apple is a fruit. So therefore, my conclusion is that this is equal to a fruit. So that's deductive reasoning. We're taking a general idea, we have a specific case, and we're applying that general idea to our specific case. Inductive reasoning, it does the opposite. Inductive reasoning takes a very specific case. I break out when I eat peanuts, so here's our specific case. And it applies that specific case to a broader understanding or a more general principle. Breaking out into hives when you eat something is a symptom of being allergic, so that's our general statement. So in inductive reasoning, I'm taking this specific statement, I'm breaking out in hives when I eat peanuts, and I'm applying this general principle. Breaking out is a symptom of being allergic, so that I can make this conclusion, I must be allergic to peanuts. So we're taking a specific, applying a general principle, making a conclusion. Another example, bag. I'm going to draw a horrible bag here, but here's my little bag. And I have a bag that I'm pulling, and you're seeing that every time I pull a coin out, I'm pulling out a penny. So I'm pulling out a 1. I pull something out of the bag. Okay, so we have 1 cent, 1 cent, 1 cent. So that's my specific. I pull something out of the bag, I'm pulling out a penny. We can apply a general principle that, well, the bag is full of something. If I'm only pulling out pennies, my conclusion could be that the bag contains pennies, contains only pennies. So I'm using a specific case, pennies are coming out of the bag to make kind of a general conclusion, the bag contains pennies. So we use both deductive and inductive reasoning, but really we'll see that we use inductive reasoning more commonly when we use the scientific method, at least in this class. So going back to the scientific method, let's actually go through kind of the process of how we start, how we complete an experiment, and how we use our experimental results to reach some type of conclusion about the question that we initially asked. So we're going to go through basically this infographic, and you can see that all science really starts here, where we are going to make observations and ask questions about our environment. So all science exists with a scientist or you noticing something about your environment or your area of study and then asking a question about it. From here, after we make these observations and after we make a question based on what we observe, we can then start to do the next step, which is formulating a hypothesis. So as a definition, a hypothesis is a testable prediction or statement that allows us to answer our question in both a precise and accurate way. These are really important. We want the data that we get from our experiment to be precise and accurate. And so some things that we should keep in mind when we make a hypothesis is, first off, a good hypothesis should always be based on what is already known about our broad area of study. So typically what this means is that we usually have to do some research so that when we ask our question, the first thing is to say, okay, well, what is already known about what I'm trying to answer? So we need to kind of start with what already exists so that I can start to make this prediction. We'll see that a good hypothesis will then allow us to make a very specific prediction. That prediction can then allow us to design a controlled experiment. We'll talk about what that means to be controlled. And that experiment's going to be kind of all based on that prediction that we make. And really importantly, a hypothesis is going to allow us to collect quantifiable data. That means that you can count that data. So we have two types of data, quantifiable and qualitative. Qualitative data is data that's kind of like yes or no, blue or purple, red or gold. It tells you about the quality of kind of the results that you're getting, whereas quantifiable data, this is what you can physically count. And we really are specifically trying to get measurable data, countable data, and that's going to allow us to determine if we should accept or reject our prediction. So let's practice this a little bit. So I have two questions for you guys. So this infographic from your textbook is really nice in that it gives this question of, does eating oatmeal lower blood cholesterol? And then we can make this hypothesis that yes, eating oatmeal lowers blood cholesterol levels. So here's kind of your example hypothesis that can then allow you to come up with a prediction. If oatmeal consumption lowers blood cholesterol levels, then a person's cholesterol level will be lowered by eating a bowl of oatmeal a day for 6 weeks. Notice that the hypothesis is fairly broad. Okay, we're making this statement that yes, eating this thing can lower this characteristic about your blood. But the prediction is very specific. Notice that it's kind of already telling you how this person's going to set up their experiment. We have oatmeal consumption. Notice that if oatmeal consumption lowers blood cholesterol levels, there's their hypothesis, again, kind of in this if statement. The then portion of this prediction is very specific in that it says someone's going to eat a bowl of oatmeal every day for 6 weeks, and that will lower their cholesterol level. So our prediction kind of takes our hypothesis and makes a very specific statement about what the experimenter or the scientist hopes to see. So let's think about question number 1. Do Formula One race car drivers have faster reaction times than the average person? Okay, well, so with that, if I'm kind of sticking with the example that I've given on the left, my hypothesis can be fairly broad, that yes, I can just say Formula One race car drivers do have faster reaction times than the average person. But notice that when I make my prediction then, so I have my broad statement for my hypothesis, Formula One drivers do have a faster reaction time than the average person, but my prediction is very specific. If Formula One drivers have a faster reaction time, then a group of drivers' reaction times can be measured and compared to a group of average people's reactions. So I'm saying that we can put these two people in the same situation and we can compare their reaction times. Similarly, for number 2, does regular exercise lower a person's body fat percentage? I can make a general hypothesis that yes, regular exercise does lower body fat percentage, but my prediction becomes very specific. If regular exercise does lower body fat percentage, there's my hypothesis, then a person will show a decreased body fat after performing a regular exercise routine over the course of 8 weeks. So notice that this last portion of my statement is very specific, that I'm having a regular exercise over 8 weeks and then I'm going to measure body fat percentage. You'll have some more practice with this as we kind of go through week 2, especially with your lab exercise for week 2. But the thing I want to point out is that notice that we're always starting with a question. Our hypothesis basically turns that question into a statement, a broad statement, and then we can use that to make a very specific prediction. And importantly, that last statement, this section kind of that I'm circling, that is testable. That means that we could actually design an experiment around that prediction. So now we've asked a question, we've formulated a hypothesis, we've made a prediction. The next part of our goal in the scientific method is to set up a controlled experiment. So this is now where we are going to test our prediction. To do that, we need the following things. First off, we need what is called an independent variable, or you'll often see me shorten this to IV. The independent variable is going to be the aspect of our experiment that we manipulate. We are scientists, you are a scientist watching this video, and you are going to change something. In our example, of cholesterol and oatmeal, we would be changing whether the person eats oatmeal or not. With our race car drivers, we could change whether someone is a professional driver or a normal driver, or whether someone has regular exercise or doesn't exercise. We are controlling that thing in our environment. When we have an independent variable, we're then going to measure something. What we measure is called the dependent variable. This is what we are going to actually count. You'll see me shorten this as DV when we collect our results. So for example, what is the cholesterol level after eating oatmeal? What are the actual reaction times? What is the body fat percentage after performing or not performing exercise? But in all of these things, we are going to collect quantifiable data that we can count so that we can measure how that independent variable affects or acts upon the dependent variable. So we'll always see that the IV is always acting upon the DV. So you can think the dependent variable depends on your independent. I always also like to remind my students to think of the I in independent variable as helping you remember that I change the independent variable. And then from here, when we change, whether someone's a professional driver or not, whether someone exercises or doesn't, or whether they eat oatmeal or doesn't, we need to have 2 different groups in our experiment, and those are called the control group and the experimental group. The control group is a group of people or test subjects who do not receive your independent variable. So these are going to be people who don't eat oatmeal or who are considered average drivers or people who don't exercise. They are not receiving the thing that you are trying to change. Your experimental group, on the other hand, will receive your independent variable. There are some things to keep in mind, though, when setting up these 2 groups. So again, sticking with does oatmeal consumption lower blood cholesterol? If we were to set up our control and our experimental groups, this is what they would look like for this experiment. Remember, our independent variable is oatmeal for this example. That's the thing that we would change if we were the scientists here. And the dependent variable, the thing that we measure, is blood cholesterol. So our experimental group, that group of people is going to get oatmeal every day. So they receive the independent variable. But notice that on the right-hand side, so it's not ethical to just not feed people. So in this experiment, notice the control group, these people are still eating something, but they're eating farina. It's a different type of grain. So they're not receiving oatmeal. They're not receiving your independent variable. They're receiving something else to eat. But those would be our two groups for this blood cholesterol experiment. Some things to keep in mind though when we set up those groups. First off, who goes into which group, the control or the experimental, needs to be random. So you're not going to say like, oh, well, only women will be in the control group and only men will be in the experimental group. You're really just going to have a group of people and you randomly pick their name out of a card and say, okay, you're in this group, you're in that group. Between the two groups, your sample size should be relatively the same. That means if there's 100 people in your control group, there should be about 100 people in your experimental group. The other really big thing, and this is a really key idea to keep in mind when setting up controlled experiments, is that the variable that you are manipulating, your independent variable, that is the only thing that should change, that should differ between the two groups. Everything else has to be standardized. So these people, the only thing that can be different is what they are eating. Their like levels of activities, their kind of what they do day to day. You need to try to control everything else to relatively the same. Everything else needs to be standardized. Another kind of thing to keep in mind, especially with your sample size, is that in general, the larger your sample size, the more accurately it will represent the entire population. So the bigger the group, the better. 100 people is pretty small. You actually usually want thousands of people. To give you guys an idea or an example of that, the COVID-19 vaccine, that had, so the vaccine study, particularly for the Pfizer vaccine, it tested about, I'm just going to round this up to about 48,000 people. And they came from all different backgrounds, so all different races, all different ages, and those groups were roughly split into two groups of 24,000 people. So they roughly split those groups randomly in half. Half of those people received the vaccine, half received a placebo. So this is a pretty good sample size, and they also made sure that of the people that were in these 48,000 people, that it represented kind of the demographics of the United States. So we had, a certain percentage were white, and that matched, for instance, the percentage of the entire population that was white. A certain amount was African American, that represented the same percentage that we see in the entire population, and so on and so forth. So we have white, black, Asian, we had Hispanic, we had Native American. So they really took into account all of the different races, demographics that we see, and they tried to make those look as much like the entire population as they can to say that, look, we basically made a miniature United States in this experiment so that our data is accurate, so that it accurately describes how all people in our country would react to this vaccine. Okay, so just to kind of recap, we have made an observation, we've formed a question, we've come up with a hypothesis that then allows us to make a very specific prediction, and we have set up an experiment, a controlled experiment that will allow us, in this example, to test if eating oatmeal really does help your blood cholesterol. And then basically what these people are going to do is they're going to collect results. They will measure the blood cholesterol that is seen in the control group and the experimental group's blood, and they'll use those results to make a conclusion. So let's actually look at how we collect data and how we make conclusions to kind of wrap up our discussion about the scientific method. So typically, once we've performed an experiment, we're going to collect our data and we're going to analyze our results by putting our data into graphical form. We can also make tables, but graphs are really nice in that they very clearly show if there's a visual relationship between what we were testing. So notice that in this situation, we have a title of our graph which tells us the effects of oatmeal diet on blood cholesterol. Typically, the independent variable goes on the x-axis, the axis that goes on across the bottom of our graph, and then our dependent variable goes on the y-axis. So notice we have blood cholesterol levels measuring here on the y, and our two groups on the left. I'm going to assume, because your textbook didn't give a key, that purple is kind of before their blood level, their blood cholesterol before this oatmeal diet, again, before the treatment, and then blood cholesterol after is in yellow. So when we look at this, notice that, remember, our control group, they did not eat oatmeal, they ate a different type of grain. But notice that with our experimental group, the group who did eat oatmeal, we see a pretty big difference between their blood cholesterol levels before and after, whereas that difference in our control group is not very big. So we can see that there does appear to be a measurable difference between with eating oatmeal and lowering cholesterol levels. So from there, when we have this graphical form and once we've analyzed it, we can then make a conclusion. So from here, would you say that these results, these accept or reject our hypothesis? Remember, our hypothesis was eating oatmeal reduces cholesterol level or changes cholesterol level. Is that true? Yes. So I would say that our results allow us to accept our hypothesis that having an oatmeal diet does have an effect, and specifically a lowering effect on blood cholesterol levels. So what's cool here though, and I want to kind of go back to the previous infographic, so notice that, so now we have kind of this idea that yes, we can lower blood cholesterol by eating oatmeal. But from here, we can actually ask more questions. Science truly is never ending. So we can use those results to say, okay, well, how much oatmeal do you actually have to eat to see a lowered cholesterol level in your blood? And so we can use those results to ask yet another question and perform yet another experiment. So for instance, maybe after we've accepted our first hypothesis, we can then ask, okay, well, let's talk about the actual amount of oatmeal that's required. And we can say, well, now we can make a new prediction that if one consumes greater amounts of oatmeal, then the lowering effect should be greater. And now if we perform that experiment, notice we have multiple groups. We still have our group of people that ate farina, so that's still in orange, our control group. But now we actually have three experimental groups. We have a group in purple, I'm just going to label them as experimental number 1, who ate only 1 ounce of oatmeal a day. We have a group in green, we'll call them experimental group 2, that ate 2 ounces of oatmeal. And we have a third group, experimental group 3 in blue, that ate 3 ounces of oatmeal. And so when we look at these results, now notice that we can make kind of even stronger conclusions where it looks like the amount of oatmeal that you eat does matter. For instance, we see that the people who ate 2 ounces of oatmeal every day seem to have, on average, a much lower blood cholesterol level than people who ate 1 or 3 ounces a day. But in both cases, we can see that no matter what, when you eat oatmeal, that's always lower. Your blood cholesterol is always lower than when you ate farina, that control group. So we can really use the results that we get from our first experiment to refine and ask even more specific questions to get even kind of a stronger understanding about how oatmeal, for example, affects blood cholesterol. So why is this important? Why do we need to have the scientific method? Why do we need to perform experiments in a controlled way to get accurate and precise data? Well, really it's because of this. When we perform experiments, the whole goal is that the answers that we get from our experimentation allows us to make theories. Typically, multiple scientists are working simultaneously to answer multiple different questions within an area of study. So maybe there are groups that are looking at how oatmeal lowers your blood cholesterol, but maybe there's another group that's looking at, well, why does blood cholesterol need to be lower in the first place? And so what's really important is that the repetition of experimentation, so multiple people could be performing the same blood cholesterol and oatmeal experiment, for example, plus having a variety of an experimentation, multiple people are looking at the importance of blood cholesterol for human health. Having both repetition and variety allows us to create scientific theories, which are basically broad scope statements about our physical universe. What's really important about theories is that they can be modified. As we learn more about those specific topics, we can adapt the theories, we can evolve the theories to fit what we know better. And sometimes theories can be completely rejected. This especially happened with scientists who were studying evolution back in the day. There were multiple different theories to help explain how organisms change from one generation to the next. And it was eventually found that Darwin's theory was true, and so the others were thrown out. Another kind of going back to the 7 characteristics of life, there are what is called the cell theory of life, can't spell theory, which stays shown in this infographic. So this is the cell theory. So cell theory states that all living things are made of cells. So notice that was kind of one of our big characteristics that's shared between all living organisms. Cell theory has also shown us that cells are the smallest units of life. They're truly considered, you have to have a cell in order to be considered a living thing. And we now know through experimentation that cells come only from other cells. So you can't just, a cell just isn't going to appear out of nowhere. You have to have a cell that can then divide to reproduce a new cell. So theories are broad scope statements that allow us to understand our universe. Cell theory is a theory that allows us to better understand cells, for example. So that's really what I want you to know for both the basics of life and the scientific method. I highly recommend that you come back to this lecture when you get into week 2 for studying or for working through lab 2, which is all about the scientific method. Something else that I think can be really helpful as we get into lab 2 is for you on your own to check out chapter 1.5. This really goes through how to think critically about science, especially the information that's presented to you in research papers and things like that. I'm going to go over in more detail during week 2 on what you need to do to complete your lab report that's required to get points for lab 2. But this section of your textbook can really help you with finding primary literature and thinking about what you're finding to help back up your statements. So that is it for this video. Your next videos I will post to D12 and you can watch those on your own time as well. Thanks.